few of the slides we completed last class. No, I'll just as a recap, I'll just go through all the slides quickly. What we have done in the last last class. Then detail you will see the next one. So food poisoning, as I told, like a very common thing which usually happens because of infected ingestion of the contaminated food. And also there is a lot of epidemiologically we can see that very commonly this kind of food poisoning can occur many times, outbreaks. There are two types: foodborne infections and foodborne intoxications. And you can see that foodborne infections is because of infective microorganisms, intoxication is because of toxins. The foodborne, we see that mainly contaminations of microorganism entering into the GI tract, can be fungal, bacteria, viral, or parasitic. And there are a lot of different diseases caused by this each kind of the organisms. And last class we have seen Salmonellosis, Campylobacteriaceae, and E. coli. I think from now we have to start with the Shigellosis and other fungal, parasitic, and viral also. The Salmonellosis, you can see that a lot of organisms which causes. So transmission by a lot of contaminated food from other thing, and the clinical feature is almost common. In severe cases only it causes septicemia, leukopenia, endocarditis, and pericarditis. And the second disease is the Campylobacteriaceae, is again caused mainly by the Campylobacter jejuni and coli. Okay, and it's mainly occurs in cattle, feces, and poultry as a normal flora that can get infected to the human being. The clinical features are almost similar, and transmission is mainly by poultry, unpasteurized milk, and the whole thing. And the next disease we were seeing like E. coli, foodborne infection. So we had a lot of foodborne infections and the strains which causes different infections, like we call it as enteropathogenic, enterotoxigenic, enteroinvasive, and enterohemorrhagic. So enteropathogenic is again having two toxins like labile toxin and stable toxins, but it is not invasive type. Just a superficial it causes by contaminated food and water. The clinical feature is almost similar like other any food poisonings. And also the toxigenic, it also causes traveler's diarrhea, people traveling in different parts of the country. And enterohemorrhagic is also this one which is little bit of severe kind of reactions can cause here. So virus cytotoxin and sugar-like toxin. Okay, and also a lot of complications also it causes, and bloody diarrhea, ulcerative, acute ulcerative or ischemic colitis, and also HUS, hemolytic uremic syndrome also it causes. I think we stopped here, no? Last class. Are you all there?
Yes, sir.
So I gave some questions, no, last week.
Yes, sir.
What was that? Hello?
You asked to explain about enteropathogenic, enterotoxigenic, enteroinvasive, and enterohemorrhagic.
Only that one I gave a question?
Yes, in toxicology only that.
Okay, what is enteropathogenic? What is enteropathogenic?
tending to produce disease, tending to produce disease in the intestinal tract.
Hmm. Okay. Only to the restricted to the intestinal thing. Enterotoxigenic.
Toxins produced in the intestinal region to produce diarrheal disease in human animals.
Then only one person is telling her. What about others?
And sir, enterotoxigenic is a type of E. coli and one of the leading bacterial cause of diarrhea in the developing world, like as well as the most common cause of traveler's diarrhea.
Okay. Then
Then
It
enteroinvasive is
uses colonization factors.
okay. Then
enteroinvasive. It is a type of pathogenic bacteria whose infection causes a syndrome that is identical to Shigellosis with profuse diarrhea and high fever.
Fever will be
Microinflammation is
around
invasive to
38.5
epithelial
degrees
cells.
and it can last to 5 to 7 days. Like usually they get better in 5 to 7 days.
okay. Next, enterohemorrhagic.
Enterohemorrhagic is one of the many group of bacteria that normally live in the intestinal or healthy human and most warm-blooded animals like E. coli. E. coli bacteria help to maintain the balance of normal intestinal bacteria against harmful bacteria.
Okay.
Infection in the intestine that causes hemorrhage within the intestine is called enterohemorrhagic.
Causes infection within the
Infection in the intestine that causes hemorrhage within the intestine.
okay. I asked now what is the hemolytic uremic syndrome also, HUS?
Yes, sir. Yes, sir.
Drugs causing HUS also is there.
Asked that time and someone answered, sir.
Yeah, you asked me what is that risk
That
EHS
time only
infection.
you
That
asked
there are
and
many.
answered.
I only told answer.
The destruction of the blood platelets. Hello. It causes anemia, so usually. Hemolytic uremic syndrome.
How it causes? What is the mechanism? It is a very good mechanism, no? I will explain that.
Yes, sir.
Sir, toxin damage to the lining of intestinal wall. So this is an effect to the flora and that causes to the anemia.
No, HUS I am asking, hemolytic uremic syndrome. Hemolytic uremic syndrome.
It produces Shigellosis toxin, which damages the lining of the intestinal walls.
So what happens in HUS, hemolytic uremic syndrome? Which organ
The
is
RBC, it spreads. So as the
Hemolysis will occur, sir, and that we can find in the urine. That's why it is called uremic.
Okay, so it is even the hemolysis and urea increases. Actually, it affects the kidney more than the intestine. Hmm? Okay, so many of are not involved in the daily discussion. Hmm? Only just they log in and just keep quiet. I think we have to give some assignments to them. Okay, so we'll go to the continue the next slides. Up to the last slide we done in the completed in the last class, and this is the new information. So Shigellosis. So this is also known as Bacillary dysentery, which was used to quite common earlier, and a lot of different genus can cause this: Shigella dysentery, Shigella flexneri, Shigella boydii, and Shigella soni. The very small amount of organisms also can cause this Shigellosis. Maybe even 100 to 10 to 100 bacilli it causes. And spread is by fecal-oral route. Quite commonly, this is the one of the very quite common route. Illness begins 1 to 4 days after ingestion of bacteria and may last 4 to 7 days. The symptoms include like fever, same like any other food poisoning, watery or bloody diarrhea, nausea and vomiting, abdominal pain, and dehydrations. The next bacterial infection is cholera. It's also caused by Vibrio cholera. So, okay, it is ingested in again water or food, and it will multiply in the small intestine to produce very potent enterotoxins, which stimulates a persistent outpouring of isotonic fluid. So it does not allow the fluid to get absorbed. So that is one of the most common issue with cholera. So the intracellular fluid all will get excreted to the gut, GIT, and get diarrhea. Okay, so that is what outpouring of isotonic fluid by the gut mucosal cells. So it's also an enterotoxins which causes, and is again transmitted by the host of the cholera vibrios from person to person through contaminated water or foods. Actually, even if you go back like 40, 50 years, like how now corona issue is there, everything is locked down. So that time, if cholera used to come, they used to whole village used to village or place used to get vacate because a lot of people used to die just because of cholera. But now people have become resistant to it and does not to worry. But that time it was a very serious disease. Like shrimps, contaminated water, shellfish, and vegetables are the most common carriers. Symptom is again vomiting, diarrhea, and 20 to 30 stools per day. Just see that frequent loss of water occurs. So almost 20 to 30 times in a day. So this is makes the sudden depletion of water in the body. So when there is a sudden depletion of water in the body, as a hypovolemia occurs because of the decreased volume, and it can cause cardiovascular complications, renal, and also cerebrovascular perfusions can impact. So death may occur in 12 to 24 hours due to diabetes and also hypovolemic shock. And Vibrio parahemolyticus, it's also pathogenic bacteria by foodborne. It's mainly through sea. It comes this one through the sea, where you can see that fish is one of the source for this. If you see here, the humans occur mainly for seafoods such as oysters, shrimps, crabs, lobsters, fish, clams, and related shellfish. Okay, so this is also Vibrio parahemolyticus. It's also in gastroenteritis damage it causes and extraintestinal infections in man. The incubation period is 16 hours. Also, symptoms is quite similar, almost like any other thing. And also, Vibrio vulnificus, foodborne infections are there, which cause severe foodborne infection associated with the oysters, eating raw oysters. And also, in immunocompromised patients, Vibrio vulnificus can cause fever, volume, myalgia, and abdominal. Incubation period is between 24 to 48 hours. And also Yersinia enterocolitica. It's also organism isolated from beef, lamb, pork, seafoods, vegetables, milk, and cakes, and also vacuum packed meat. So this is a source of mainly through the flesh, okay, of other animals. So symptoms are same. The organisms mainly steady shoots up to 40 days following illness. So it has a chance of causing infection in another person by almost for 40 days. So that's all about the bacterial borne infections. Now foodborne infections. Now viral foodborne infections. So usually they are again transmitted through food. Like hepatitis A and Norwalk-like virus, like Norovirus. They are the most common foodborne disease infections, and this may spread very widely. That's the thing. So always we say like foodborne, the virus spreading is so quick. Like hepatitis A, it's also a very long period incubation, like 30 days it may cause the incubation period. Signs and symptoms are almost similar: fever, muscle aches, headache, loss of appetite, abdominal discomfort, weakness, and fatigue. And the other one is Norwalk-like virus. It's a very mild one infection. Incubation period is of 3 days. So consumption of raw oysters, clinical manifestations include vomiting and diarrhea, and very rarely it can cause convulsions also. So till now we have seen foodborne infections. Now we will see foodborne intoxications. So I told very clearly difference between infection and intoxications. Infection is nothing but causing direct effect by the microorganism, pathogenic microorganisms. Intoxication is nothing but it is because of the damage caused by the toxins released by the microorganisms. So, see, my voice is clear?
Yes, sir.
Yes, sir.
Okay.
Yes.
Yes.
Okay, thank God. Again, no problem. Huh? So, foodborne intoxications, again, caused by consumption of food containing biotoxicants, metabolic products, or poisonous substances. So, biotoxicants means somewhere it's found in plants and animals tissues. Some like mushroom toxicity, very common mushroom because it has a particular toxins which can cause may not be accepted by human body. Okay, and also like metabolic toxins like something which is formed or excreted by microorganisms. So in the food or in the GI tract of a person, during that metabolic product of microorganisms can become a toxic to the human being. And also the poisonous substances which are present in food during production, processing, transportation, or storage. So this process it can cause the foodborne intoxications. And also very short incubation period is there here because unlike like microorganisms, the incubation period can be go up to 14 days, 30 days can occur. So here the toxins will have a very direct effect because it's a ready-made compound toxins. and because it are characterized by lack of fever. That is the another thing, important difference between these. So again, classification is done as like bacterial, fungal, chemical, plant, and also poisonous animals. So in this way, these intoxicants are. So in bacterial intoxicants, we have Staphylococcus aureus, Bacillus cereus, Clostridium perfringens, and Clostridium botulinum. So in this Clostridium botulinum is very poisonous. Sometimes Staphylococcus also. So they causes may present in the food or any kind of things which causes the toxicity to the human cells. So like Staphylococcus aureus, they are contaminated with food. They produces enterotoxins, certain types of Staphylococcus aureus. Like the enterotoxins, there are 5 types: A, B, C, D, E. Okay. So these enterotoxins are nothing but which are released by these microorganisms which causes toxicity. So these are individual strains of may produce one or more enterotoxin types while growing in food. So you can see that how the toxins productions happens. So when it is in a temperature of 12 to 44 degrees Celsius at pH 4.2 and salt concentration of less than 10%, only these toxins here it is. You can see that salt, salt also is mainly like avoids the organism's growth. You can see that in homemade oil they put the pickles. So when you want to prepare the pickle, they put lot of huge salt. So that itself is there. So you can see that if higher concentration of salt is there, the organism the toxins does not grow. That's why pickles can be stored for so long, even though they are raw. And see, these toxins cannot grow in salt concentration of less than 10%. And if it is any temperatures below 12 degree, there is no toxin production, pH less than 4.2 or more than 10%. So that's why we keep the foods, all those things in the refrigerator, which does not allow this one to grow toxins to produce. The nature of enterotoxins, so they are heat stable, Staphylococcus in this. Ordinary cooking procedure, pasteurization or drying do not activate. Unlike like other toxins, this will not, by just cooking, drying, do not activate this. They are also pH stable and resistant to most proteolytic enzymes like trypsin, chymotrypsin, renin, and pepsin. Foods like milk products, like pasteurized milk, yogurt, meat products, fish, shellfish, poultry, and non-meat products, egg products, vegetables or cereals can cause this. Also, incubation period is very quick. You can see that 1 to 6 hours. So if there is at least 1 microgram of enterotoxins, the concentration required to cause pathogenic. And also, the signs and symptoms are almost same. Sometimes diarrhea with prostration, as a prostate gland, this one. And also, duration of illness is 24 to 72 hours. That's all. The other intoxication in bacteria is Bacillus cereus. They also caused by enterotoxins by Bacillus cereus. Here, it causes 2 diarrheal enterotoxin like hemolysin enterotoxin and non-hemolytic. The other one is the emetic toxin. Bacillus cereus is caused again by the most of the time non-vegetarian and the dairy products, meat, eggs, dairy products, cereals, dishes also may cause here poisoning. And as I told, there is emetic and diarrhea, 2 types. One is in emetic syndrome, nausea, vomiting, abdominal cramps, same, and rice, pasta based foods are very common involved. And diarrheal syndrome again, profuse diarrhea, abdominal cramps, tenesmus, rarely vomiting, and fever is absent here. Only diarrhea, loss of fluid is more. Like meat, vegetables, dried beans, and cereals can cause more of the bacillus diarrheal kind of symptoms. Are you following?
Yes, sir.
Yes, sir.
See, these are
Sir.
all very
Yes,
most
sir.
common thing. There is not much more because you may be aware that, but because chapter is involved, all these things, I'm just keep moving. Is that okay? No, you're following?
Yes, sir.
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Yes, sir.
So, foodborne intoxication also called the Clostridium perfringens, enterotoxins, CPE. So also it's a strain caused by perfringens. This is found in mines of soil, dust, water, sewage, marine sediment, decaying materials, and GI tracts. And also through a lot of contaminated food only. This is caused. Again, there are three types: A, C, D. Okay, so I don't know why no B is there. Only A, C, D is there, and heat resistant and survive heat up to 100 degree for 1 year, 1 hour. So that's the problem with these toxins. So even if you cook, boil anything, it may not usually destroy. But the concentration at 10 to the power of 7 to 10 to the power of 8 cells per gram of food, if it is there, then only this infection is caused. The causative foods is again red meats, chickens, fish, pork, fruits and vegetables. And also some mainly people who are in the slaughterhouse meat containers like handlers, dust or water, they get contaminated there. That are prepared one day and served the next day. I think this is quite commonly thing. But even if it is served next day, it has to be heated properly and served. So even the eating of such food is inadequate to destroy heat-resistant endospores. If it is heat-resistant endospores are there, even if it is eating also, it will not be. Again, 8 to 24 hours after ingestion, the incubation period. Same like nausea, intestinal cramps, and diarrhea causes. And vomiting is rare, and illness may take duration of 1 to 2 days. The next is the Clostridium botulinum, foodborne intoxications. This also, as I told, it's a very important thing. It causes a lot of complications thing. It's also caused by Clostridium botulinum enterotoxins. Also have different strains A, B, C, 1, 2, D, F, and G. They can, why it was this one is because they can sustain up to 120 degrees Celsius. And like type E strains are like non-proteolytic, while the rest are all proteolytic. Means enzymes can break down this one protease enzymes. So usually occurs at temperature 25 to 30 degree. And proteolytic strains produce an active botulinum toxin, while non-proteolytic strains produce inactive protoxin that require activation by trypsin. Like non-proteolytic strain, inactive protoxin, which is caused by the active toxin by the trypsin. This is what proteolytic enzyme. These are all neurotoxins. That's what I told. Like these are very very dangerous toxicity. The botulinum toxicity, highly toxic at heat level. Like till now, what all the different infections we have seen. So this is more severe because it's a neurotoxin. It also resists the action of gastric and intestinal juices. And also, it's botulinum toxin is one of the lethal poisons we use as in the biological war also. Okay, botulinum, same like anthrax they used to use. Same like botulinum also they can able to use this. You can just see that lethal dose itself. You see like 10 microgram, just microgram from a small powder they can make, and so small quantity it can become lethal dose. Patient may die, person may die. So what are the types of food implicated in here is anaerobic conditions, especially such as spoiled, canned meat, hams, bacon, stacked without hair access. So this is a thing where because the processed food, no packed meats and this one, it's very dangerous, especially in the malls and this one. Don't know how they're kept and whether the temperature is maintained or what. Sometimes power goes off or something. Okay, so that's the thing. So uncooked fresh foods are safe, but they're eaten before the toxin had time to develop. The adult botulism, it's causes incubation in men, 12 to 72 hours. Signs and symptoms are similar. Like you can see here, nerve paralysis. It's a very, because it's a neurotoxin, it causes specifically this one. Headache, dryness of skin, respiratory failure, lack of fever, or double vision. Duration of illness is just 1 to 10 days. And in the infants also, this botulism toxicity can see, especially when less than 1 years of age. This is, you can see that it will be there in the honey. So spores in the honey. So especially honey they use for the children's the food process. So we should be very, very careful in this, because the spores that is there in the honey can germinate in the GI tract with the toxin production, and that too in the infants it can be very dangerous because acute phase of the disease. Constipation, poor feeding, lethargy, weakness, and floppiness can occur. And fungal intoxications. You can see the bacterial over fungal intoxication now. And these are all mainly metabolites, which is called as mycotoxins. Myco itself, we can say like for the fungus thing, while growing in food. And the grains like poor dry storage, oil seeds, fruits, and vegetables are mostly involved. And also, it's a significance to public health is aflatoxicosis. This is a quite common thing which is caused a severe part of the the uh, fungal infections. And the another one is the fungal infections can lead for many other important complications, which can cause this production of these aflatoxins. So it's caused by aflatoxins produced by fungi like Aspergillus flavus and Aspergillus parasiticus. So there are 4 types: B1, B2, G1, and G2. 4 strains, and this can be lethal, as I told. It can cause a lot of damage to the GI tract and other parts. And because of this damage and irritant to the cells and tissues, they can cause hemorrhage, bleeding. So that's what acute hemorrhagic syndromes are most common here. And in the that is large doses. In the medium doses, is sub lethal changes can occur, like histotatic, means cells toxicity. But in a very small doses, but for a prolonged period, it causes liver tumors. It also can be potent carcinogens. The next type of intoxication is the chemical intoxication. So it's also consumption of food containing poisonous chemicals. So this can be intentionally or unintentionally can add it to the food. Okay, many times you can see in the society it can occur. So as a result of producing, processing, transporting, or storage. So there are chemical substances a lot of involved here. Heavy metals like antimony, mercury, arsenic, fluoride, lead, cadmium, cyanide, etc. And pesticides are insecticides which are used for the agriculture process, and herbicides and fungicides or radionuclides like radium, barium, iodine, and isotopes. Any such of chemicals which are mixed in the food can act as a poison. So it's a very short incubation period because they are very potent. Same like intoxicants, there is a toxicity, toxins are there. Same similar like here, from few minutes to hours, it can cause the toxicity after ingestion. So mainly involved GIT and CNS, nausea, headache, convulsions, GI irritations, abdominal cramps, vomiting and diarrhea, pallor, cyanosis, and also the blurred vision, the white commonly seen the reactions. And the next class is the biotoxicants. Biotoxicants again, it's a part of a plant or animal toxicity. So is any kind of poisonous substance is present in the plant or animal. When it is taken, ingested, the poison or toxic substances in that animal or plant will cause the this one. So it is because of metabolic activities of this. If you say animal biotoxicants, again poisonous animals consumption. One of the example is puffer fish, delicacy of China. I think even this coronavirus, you may be aware that it has come through the fish seller in Wuhan. Okay, so that was the one lady was first diagnosed with corona. Okay, could be even fish. It's not the source of fish, but it's getting there. But in this toxicants, it can cause the fishes can cause this one. And also diagnosis is specimen collection like feces, stool, leftover food or vomitus. Any kind of this foodborne infections, these specimen collections are these are all the few things which commonly collected. And microscopy, they see under the microscopy and see the organisms or ova or cyst of parasite. See the weight mount, hanging drop, and staining. These are the few techniques that you use for the separation or identification of the organisms. And also culture is very important to see the what organism they grow. Selective, non-selective medias are used according to the suspected organism. Like different Salmonella, Shigellosis, they use XLD and HEA, SSA, the Wilson Blair, Buffer glycerol. These are all the few different cultures they use for these infections. For Vibrio color, alkaline peptone water, thiosulfate secreted by sucrose or carry blade. And for Staphylococcus aureus, mannitol salt agar or milk salt agar they may use. For Bacillus series, mannitol egg yolk, phenol red polymyxin agar. Also PEMB, polymyxin B, egg yolk, mannitol, bromothymol blue agar. Okay. And also the anaerobes like RCMB, Robertson cooked meat broth, and thioglycolide broth. And also Campylobacter, Skyros, Bacillus, and Campi. So are the few cultures and different things they use for any kind of food poisoning. You can see the food poisoning here, microscopy, culture, serology, and PCR, polymerase chain reactions, very thing. And in the microscopy, light and electron, again we see parasite, hanging drop, gram stains, alien stains, virus we can see. And in the culture, stool or tissue culture, stool again direct enrichment selective. In direct McConney culture, and enrichment selenite, FP, tetrathionate, alkaline peptone, water. Also Wilson Blair, TCBs and XLDs and DCS. Identification, serotyping, antibiotic sensitivity, and biochemical reactions can be seen. Serology is very specific, quite commonly. They can do the assay and ELISA, relative or electrophoresis, they can do it. These are all the serological diagnosis. And molecular techniques, polymerase chain reaction, as I told. So if you see that overall, how you manage the food poisoning, the prevention is one of the very important for any kind of this such kind of hygiene related infections. So we should have washed the hands very frequently using restroom, shaking hands, and before eating. So this is a quite common best practice because hand can be touched in many places, materials, and come across meet many people, infected people. So hands are one of the most chances of infections, getting infections from other things, source of infections. Now, eat only pasteurized dairy products and eggs, It has to be cooked, right? And this one. And avoid tap water and ice if uncertain about quality. And high risk foods like creams, custards, eggs, berries, lettuce, fish, and shellfish. Undercooked fish and shellfish should never be used because the most common source it is from this source only. And the prevent food handlers from working while ill. Do not eat outside food or anything. Avoid salads and all raw fruits that you do not peel. So which is not the peeling process is not there. Okay. So they those kind of foods it has to be avoided. Eating raw without peeling. And also do not eat solid foods during vomiting and diarrhea. It can cause choking. And tea with lemon and ginger. As we know, like lemon is one of the immuno boot. It gives the immunity. Uh, uh, the thing. So definitely it can cause. It can increase the immunity. So it can be a protective agent again in toxins or organisms. Wash and sanitize all surface equipment and protect the kitchen areas and food from insects, pests, and others. I think now we are all very widely following all these things. Separate raw meat, poultry, and seafood from other foods, and separate utensils, knives like cutting board for handling raw foods, and choose foods processed for safety like pasteurized milk. Wash fruits and vegetables, especially if eaten raw, uh, because a lot of organisms can cause contamination. And do not use any kind of food before its expiry date, packed food or anything. So that's all about the food poisoning. Any other questions?
No, sir.
See, it's a very quite simple chapter. Somewhere we use some antibiotics for this kind of bacterial infections and this one. Especially use penicillins and some kind of ceftriaxones or anything. But apart from that, there is not much more treatment for this poisoning, especially gastric lavage, and this one only is more common. Okay. So it's a very small chapter. So we have done with this. It's a commonly used a lot of chapter. This one subheadings comes, so you have to read all these things. Okay, the management also.